Welcome to Study Loop.
So here we've got a little picture of an mRNA, and we have our 5' cap, we have our 5' UTR, we've got our exons right here, we've got our 3' UTR, and we've got our poly A tail. So the 5' and the 3' UTR are going to be non-coding, and then these exons are going to be the coding region. And this is where proteins are going to be made. So in this video, we're going to be focusing on the 5' UTR. So proteins that can bind to the 5' UTR are going to be called RNA binding proteins, or RBPs for short. And they bind the UTR. They can bind either one. They can bind the 3' or the 5'. And what they are going to do is they are going to increase or decrease the stability. So you have some RBPs that are going to increase the mRNA stability and some that are going to decrease the mRNA stability. So if you had a mutation in this area, we'll write an M for mutation, and the 5' UTR was no longer able to bind any protein, then you would see that the RBPs would not be able to bind. So if you had an RBP that increased stability and it was no longer able to bind, then the stability would then decrease, and vice versa for an RBP that would decrease stability. So mutation here would change the stability of the mRNA. So some things that wouldn't change would be the splicing, the ability for splicing, because that's going to occur in the nucleus, which is before the cytoplasm, which is where we're talking about here. The translation would not be stopped, because that would be, you would have to be affecting the ribosome binding site. So we'll write ribosome here, since they're the ones that are actually translating the proteins. And then also you're not going to be affecting the poly A tail at all, because as you can see, the poly A tail is way over here on the 3' end, and we're over here on this 5' end over here.